can you hear me? Professor Wang, can you hear me?
I think I did, but can you be a bit louder?
Okay, is this sounds okay?
Okay, people in the back, can you hear? Okay. Can we bring the volume up here a bit? Okay, so today we have Professor Ting from Taiwan, and she's going to tell us about CRISPR gene editing, which is really hot technique.
Okay.
Yeah, it's yours.
Yeah. Good morning, everyone. Today, we want to talk about this new technology, and I think this is one of the sort of like a well-known technology in life science, and also revolutionize a lot of our research and maybe clinical application as well. So to begin,
Professor Ting, can I interrupt you for a second?
Yes.
We cannot see your slides.
You cannot see my slides. Okay.
Is that our channel, or maybe this? Is that our project? Oh, maybe that's working now.
Is it okay now?
Oh yes, yes. Thank you.
Okay. Okay. Good. Okay. So this is a new technology. As I said, it revolutionized all life science research and probably also clinical applications. I want to start with, in the past, we utilized a lot of model organism and to do research. Then one particular reason is because we can do genetics using this model organism. And every single one here has been utilized for many, many years, especially mice, flies, and yeast. But with those, there's always a way to, or a difficulty to understand gene function. So traditional, we use mutagenesis to do and try to understand gene function, but there's a limitation for that. So ideally, using Drosophila as a model organism, or we can replace Drosophila on this slide to any other model organism. What we would like to do is to understand the biology and also understand some of the mechanisms. And in the future, we want to apply this knowledge to improve human health. That's one of the reasons we are here. But in addition to this mission, actually, we did very well in using model organism to study the biology and try to understand the function of individual genes in the past. But our goal was to understand the human biology. So mostly we want to improve human health, especially now in most all populations are in approaching aging. Then there's a lot of the issues with human health and some of the common diseases or symptoms in the population. And using model organism here, I use Drosophila, but you can replace this as a zebrafish or C. elegans, and we can think about how they are similar in terms of internal or external structure, and also at the cellular molecular level, and there are a lot of similarity. Down to the gene level, there's also conservations across a large number of species. So as a geneticist, we have two approaches. One is the forward genetics approach. The other is reverse genetics approach. Forward genetics approach usually focus on a specific biological process. So I always take an example as my colleagues study eye development. So what he would like to do is to identify all genes in the genome which involve in the eye development in a little fruit fly. And to do this, traditionally we do mutagenesis. We try to mutagenize the organism and identify mutant phenotype. Then once we have the mutant phenotype, then we ask which gene was affected, and also like how we connect all the genes together to give rise this beautiful structure. And on the other hand, reverse genetics become more popular these days because a lot of time we have the genome ready first. So we complete the human genome, and along the time, we complete many genomes across the world. Then the difficulty now is with so many genes, like tens of thousands of genes, and what are these genes do, and also, you know, what specific role they play in a single organism. So here I use Drosophila to explain, and this was a slide people utilized to explain the forward genetics. As I say, we mutagenize the organism, and we can select a specific mutant and try to understand which gene was affected. Then we connect this, identify the responsible gene, and hopefully that link the sequence of a gene and the molecular feature of the gene link to, you know, one of the interested area. But this slide can also go backwards. So the backwards is like from E to A. So if we start with the human diseases or human phenotype, so nowadays we are not only interested in, you know, disease-related genes, we are also interested in the genes contribute to this organism. So if we identify a series of genes, then we can use, because we cannot use human as a sort of like an experimental organism, but we can use mice or other, for example, Drosophila or C. elegans or some like a zebrafish. Then we can identify the homolog in Drosophila. So at the sequence level, there are over 70% of genes. We can find human homolog in Drosophila genome. So in those cases, we have the gene first. Then we want to target this specific gene and to study whether eliminate or knock out this gene will give rise to any phenotype. And we will be able to understand the gene function this way. So this slide, if we go backwards, we can also study, and that's what we mean by reverse genetics. But the problem is, how do we target the gene? And this is not a new question. Human geneticists, Drosophila geneticists, mouse geneticists, they all want to target a gene if they know the molecule. Now it's very relatively easy to target one specific gene in the genome. But molecularly, within the cell, especially within the germline, if we want to target, it's very, very difficult. And in the beginning, there's no such a system, and we cannot culture Drosophila cell for germline. We can culture Drosophila cells, so we have Drosophila cell lines, but we cannot really culture germline cells for Drosophila and do transplantation like in mice. And also, it would be difficult at that time to introduce DNA into the cell, into the embryo. So the idea is if we inject the DNA into the early embryo, and we might affect the germline in this way. But there's also a difficulty to do in a single embryo. Then there's another difficulty is the repair system. So during the early development, in Drosophila, the repair was almost stopped. So DNA repair was not available during that time. So given all those difficulties, actually in year 2000, there's a very old-fashioned new technique, old-fashioned new technique. So that's, I need to give the credit to the two scientists, Ron and Golick, and both of them develop method for Drosophila genetics to overcome this targeting issue. I want to start with this technique, although most people don't use this technique anymore, but we still use very similar machinery to target genes in not only in flies, but also in other organisms. So they try to imagine, because we want to do a germline transformation. So everything has to happen in a germline. So one thing is, they have to have donor. So the donor, you will see in a minute, is the DNA we designed, and we would like to target to the particular genome location. Then they want to use a system called recombinase. Recombinase is the enzyme to facilitate homologous recombination. But homologous recombination is sometimes the frequency is not so high. So what they did, they borrow a system from yeast. So it's called fleepase FRT system. Fleepase is the enzyme. FRT is a specific DNA sequence. When the two get together, they will facilitate recombination. So this will help us to, excise DNA loop or ectopically recombination could occur. And the third thing they utilize is an enzyme. So this is the endonuclease. And this endonuclease is similar to restriction enzyme we use in bacteria and plasmid, but this enzyme recognizes like longer sequences. So it's a rare cutter if we use the name we sort of commonly call. So this ISCE1 is an endonuclease, so they can cut the DNA in the cell. And the last thing is, for everything we do in a cell level, we want to be able to see that later in the organism. So for us, screening the fly is relatively easy than just detecting the molecular basis. So this is basically the construct they design. So here I want to explain what is here. So in this construct, they have the donors, and this donor DNA will loop out hopefully in the germline. And the way to loop it out is by this FRT site, as this in the previous slide. And this is the ISE one we use later because we want to cut the DNA to create a double strand. And the whole donor construct is not freely injected, because when they inject the free DNA into the early embryo, then they just failed every single time. So they decide to use transgenic approach. So they put the transposable element sequence on this construct. So this construct, the A construct, was a transgenic construct integrated to the fly genome. So this fly, so it's sitting in the middle, this fly already have the donor DNA in the genome in every single cell. And this is how they deliver donor to the cell. So that's something, they design. And this is where the endonuclease, so the endonuclease here is to create a double strand break. So this donor DNA, they made a cut because the donor DNA was designed. And the bottom, the purple part, the dark blue purple, that's the DNA sequence we want to target. And the light blue and dark blue are the homologous parts. Okay. So if they already know the sequence of their target, they can carefully design this donor, and hopefully in the germline, this integration will happen. And the red part here is just the marker we can follow. Okay. With this very complicated method, they were able to target the first Drosophila gene in year 2000. And of course, many people follow their strategy, but it was just like very hard. And the successful rate was not easy to estimate it. So I want to turn to what we want to do. So we want to really target the specific gene. And not only target and mutate it, we also want to engineer it. So engineering means, we want to change the base, we want to change the allele type, and at its original location. And this is not easy. It's not easy in any organism until we have some handle try to manipulate the gene by a series of different endonucleus. But the principle for this is to making the double strand break at the target site. I want to go back to this slide. So remember, the first Drosophila strategy here is making the double strand break at the donor. So the circular donor will be cut by endonucleus. Then after cutting by endonucleus, then they will have a chance to integrate into the target site. But with the series of gene editing or engineering method, we try to find a suitable enzyme, like the ISE1. But instead of cutting the donor, we try to cut right at the genome site. So this is something, we have to match the protein, the endonucleus, or the enzyme similar to endonucleus has to be able to find the target in the cell. And that's the difficult part. So I want to introduce very quickly some of the attempts people have been doing. Okay, so of course, the first thing is find the enzyme. Find the enzyme recognize a large, relatively large size of base pairs. So here we call it mega nucleus. So nucleus is just like the enzyme cut the DNA, but the mega means they recognize a large sequence size. So usually, the one we learned in molecular biology in the beginning was like 6 cutters, 4 cutters, but now it's like 20 to 40 base pairs. And of course, the recognition is rely on the enzyme. So one way to make it is more useful to target is to make only not to discover the mega enzyme, but we make a hybrid mega enzyme. So we can target many different possible locations. So that's the first category. Then along the line, people discover and we realize we might be able to use a group of protein called zinc finger proteins. Zinc finger proteins recognize DNA sequence, and they have some specificity to the nucleotides. And scientists just propose to combine the zinc finger domain, that's the domain recognize DNA, with the endonucleus. So the pink in the middle is the cutting enzyme, but they combine a hybrid enzyme, zinc finger endonucleus. So they combine it using the zinc finger to recognize nucleotide and utilize the endonucleus activity to cut to make a double strand break. So this is like a second generation. And for each finger, so zinc finger means like they have fingers, so fingers are like each small domain. For each those small domain, they can recognize 3 base pair. And you can think if we have many of this combination of domains, then we have some way to handle, variety of genome sequences. So it was popular for a while until TALEN comes up. So TALEN is another type of a hybrid enzyme. But the differences between the TALEN and the zinc finger is the small module of TALEN recognize single base. Okay, so think about if the TALEN module only recognizes single base, now you can have all different combinations. And for a small stretch of DNA can be recognized. Given the complexity in the genome, and it will be possible to target every single location you would like to do. So all these methods here is just trying to find a suitable way to cut the DNA. So we start with, the enzyme recognize a huge size, a small huge size, like a 20 to 40, to the zinc finger domain recognizes 3 base pair, to, we have module only recognize 1 base. And this is a big improvement at that time because people think now we can target every gene without a problem because we can recognize, all the sequence. We can do all kinds of combination. And this is what, we really want to do. But talent, I always say talent has very short shelf life because right after people start using talent, and we discovered this CRISPR pathway. So the discovery of CRISPR is very interesting. So in the very early days, when scientists doing some bioinformatics and evolutionary study, he found within the bacteria genome, there's a stretch of sequence homologous to phage. And this is new because we study phage and we sequence a lot of phage genome, and so we know phage quite well. And we also utilize phage in molecular cloning and do a lot of molecular biology studies. But within the bacteria genome, we don't expect to find something similar to the phage DNA. But not only that, this stretched DNA was arranged in a very different way. So it have a tandem array, and in this tandem array, the small stretch match to some of the different phage DNA or foreign DNAs. So we can trace their origin. So at that time, this scientist hypothesized this could be one of the adaptive immune system in bacteria. Okay, I have to tell you, back in that time, nobody heard about adaptive immunity in bacteria. Adaptive immunity only happen in eukaryotes, and we study human quite well, so we know how immune system work. But for bacteria, nobody has any idea about how bacteria defend. We know they have new endonuclease to defend the foreign DNA, but we never realized there's also adaptive immunity. So this was silenced for a very long time. Based on sequence, it's just a hypothesis. Not until, in the early 2000s, in early this century, people start realize the whole machinery. It turns out bacteria, this is one of the bacteria immune system, and once it's affected by the phage will insert the DNA into the bacterial cell. And there's a machinery to recognize this DNA. Then this DNA can be incorporated into the genome. Once this is done, when the second time the same phage or similar sequenced phage integrate or insert into the host cell, it already have the system. The system not only have the sequence memorized from the previous infection, but they have other machinery to collaborate together. So they have enzymatic system, they have RNA system working together. Then this short stretch red sequence from the previous infection was the memory which can target to this second infection. So the ideal situation is the red sequence can recognize the foreign sequence by their homology. And because it was carried by a whole bunch of other, for example, the enzymes and the RNA molecule. So we can do this targeting, or I should say, bacteria can do this targeting quite effectively and destroy the foreign DNA quickly. So this is how naturally CRISPR work in bacteria cell. But once we realize this, so this is a very classic basic research, understand the adaptive immunity in bacteria. But once this is discovered and realized, people think, aha, can we use the very similar system but target to any eukaryotic cells? And this is how we design, or scientists to come in, many scientific group put the effort together, and they co-discover and also utilize this system. So on the back, the gray part is the enzyme. So this is the most popular enzyme we use now. We call it Cas9. Cas9 is the endonuclease. So the enzyme cut the DNA. But with how the enzyme knows where to go and where to cut relies on this RNA molecule. So this RNA molecule has two components. And one is the guide RNA. The other is a small RNA, and sort of like can help facilitate to the incorporation of the enzyme. So now in this slide, there's a green stretch. So this green stretch, you can imagine that's the pink part in the previous slide, and that's the one from the previous phage infection. But now we can modify this sequence to our target. So we know which region of the genome we would like to target, and we can design the short stretch of oligo molecule, and we can combine it with the whole system. Then with the small synthesis, just synthesis oligos, we will be able to target this genomic region. So this is even simpler. If you have, if you cannot imagine the differences between the guide RNA and TALEN, remember we say TALEN, each small module recognizes a specific nucleotide. So for the TALEN, we are engineer the protein and rely on the protein to target a specific DNA sequence. There's one layered difference. But here, we sort of like just synthesize the DNA sequence for the complementary strain. So we make an RNA molecule complementary to what we want to target. And it unwinds the DNA. So DNA is a double helix structure. Unwinds the DNA, bind to one of the strand. And there's a blue part called pen sequence. It's very short. Then the Cas9 will cut in a small distance to the pen. And the red color here is indicate the cutting of this molecule. So this is the way we create double-strand break. But what happened after you have a double-strand break? Let's go back to your cell biology. Your cell, once your cell has double-strand break, and in the cell, there's repair system. So the repair system want to repair. We don't want double-strand break. We want to fix it. When the repair system fix it, there are two major ways to fix it. One is just grab the two ends and put them together. So this is called non-homologous end joining. The other way is try to find a template, a homologous template, then do homologous repair. So this is here, I list non-homologous end joining. The other is homologous direct repair. And I have slides to explain it. So you have a double-strand break. After you have a double-strand break, there are many things could happen. And it could, just end joining and with very small lesion in the middle, or you can cut to a long stretch and to create a relatively large deletion, or you can have another donor molecule there, and you can hopefully screen for homologous recombination. Okay, so this is a major editing method. Of course, scientists also develop ways to change only single base pair and all the variety of applications. But I want to stay with these two. So a lot of time we want to create, one thing I forgot to say, in most cases, homologous recombination has very low efficiency. So a lot of time, once you have a double strand break, more often you have non-homologous end joining. Because of this, the early application of this method is to target a single gene, try to create deletions. And this is what we try to do in the Drosophila genome in the early days. So here I want to show you a target Drosophila gene as an example. And to create a mutation, we simply just cut this region. So consists with many exons here. So this is like a illustration of the gene structure. And the Cas9 scissors is the one we can design. So we can design the Cas9 with the guide RNA and specific target these two sites. And here is the results. So I want to orientate you. We have two cutting sites. One is on the within the exon 2. The other is within the last exon. Here is the exon 6. And down here, the red color is where we expect the Cas9 will cut. And the blue line, underlying region, are the guide RNAs we design. So for each cutting site, we design one specific guide RNA. So this is a sort of more technical aspect of the application. Then down here is the results. So the results is like we create many mutant alleles. So each mutant allele, after we screen it, we can look at their sequence. So in the first species here, we have several mutants, and every single mutant has both guide RNA region cut by Cas9 and also mutated. Okay, so here I want to just try to clarify. We screen many flies. We only look at the one with mutant phenotype, which I haven't tell you is this is a gene called miniature. And based on the Drosophila literature, we know this miniature, if we target this miniature gene, we'll get a small size wing. So we use this phenotype to try this on different Drosophila species. Okay, so the first species, we generate many mutants, and in each cases, we have both regions mutated. Most of the time, they create the Cas9 double-strand break was sort of fixed by end joining. Then the non-homologous end joining create various size of deletions. In only one cases here, the mutant 19, and this 19 mutation, we have a large deletion between the two cuts. So approximately in this mutation, we have the entire region deleted. And in the second species, we also have one mutation, looks like that, and the other one has like both side cut. Okay, so this was first experiment we did and tried to create deletion, but immediately we realized this can be utilized for other type of study. So I want to show you the idea. So because I already showed you, when we give the cell two guide RNAs, and both guide RNA will work and very efficiently. We don't have any single mutation, only have one cut. And this, we are not the first one or the only one demonstrate that. There are other people doing so. If you give multiple guide RNAs, they will simultaneously target many different sites. Okay, so that's the first lesson. Then because we work on with Drosophila, and we know many tools in flies already exist. So we're thinking about designing an experiment using existing system. So here is what we think. Okay, now I want you to realize, because if this is our target, and if we want to target this particular gene, usually the difficulty is there's no screening marker. When I study the first case, I use miniature because I specifically target one of the genes. I know there's a phenotype. In the early Wrong and Golly, they did the first Drosophila targeting. They target the yellow gene. They know what's the phenotype. The fly will change the body color. Okay, but when we study an unknown gene, genes new with novel function, or we don't have any idea, and there's no way we have a phenotypic screening method. And of course, nowadays, people are pushing really hard for homologous repair. So you can have the homologous donor and put the target there. But the success rate is low. What if I only just want to create a deletion? How do I screen it? The classic method is we do PCR. We just PCR many cells or many individuals and try to find the one with deletion. And that was not very efficient, so we think maybe we can utilize some existing phenomena. So here is something we're thinking. So if we have an insertional target in front of the gene, in front of the gene means, so in Drosophila, people have been used P mutagenesis transposable elements to target gene. But a lot of time, P element was not right in the gene, but nearby a gene, especially at the 5' end of the gene. So sometimes this insertion has no effect for the gene function. Sometimes the insertion has some mild effect. And actually, within the genome, if this insertion is in between two genes, we don't know, even we have a phenotype, we don't know which gene was the target gene for this phenotype. So we're thinking about we can design a method to cut this particular molecule and just screen for loss of eye color. So we have this transposable insertion as a red because it carries an eye color gene. So all the genes with the insertion will have red color. But if we create a deletion, and this deletion remove this P element, then we'll have a revertant, a mutant eye color white. So using this method, we can screen the mutations with the existing marker gene nearby. We can also utilize this using homologous recombination. So here is an example of how we do it. So remember in the beginning, I said if you are interested in medical research, then with your study, some of you might study cancer, some of you might study human-specific diseases. So just assuming you already have a target gene you're interested. So you start with, for example, whole exome sequencing from the patient, and you identify some you need to identify some of the disease-causing candidate genes. And these candidate genes, you want to utilize model organisms, so you can take your zebrafish, take your worm, or your fly. So you identify as homolog by sequence homology. Then you can do loss of function study. So this is just one of the many examples in Drosophila we can do. So in Drosophila, we will target using CRISPR-Cas9, target this gene, and at the same time, we put in a GAL4 cassette. So this GAL4 is a transcription factor from yeast. And we can express this GAL4 based on the genomic environment. So if we replace the candidate gene in Drosophila and we replace with the GAL4 enzyme, GAL4 protein, then to be able to screen it, so this GAL4, we cannot see that, but we can hook up with the fluorescence eye color. So the 3xp3, this is a promoter sequence. So this gene will specifically express at the eye field. Then this is a fluorescent color, so we can use red, green, blue, whatever color you think it's easier for your experimental design. Then this one will be integrated together with the GAL4 system. Okay. But more than that, you can see here, we specifically put a sequence nearby. So there's a TTAA sequence flanking the marker. So in the middle, this is the marker gene. To do this is we want to remove the marker gene later. So for some other applications, sometimes because you utilize the fluorescent eye color, then the color will stay there forever. If you want to study different things in the eye field, that will interfere with your staining and other. So sometimes you might want to remove that. When you remove that, we utilize a transposable element property. So there's a transposis called piggyback. Piggyback recognize TTAA sequence. So this one, given piggyback, this whole marker region will be looped out. So you can remove this one, and you no longer have this marker gene with your target event. So this marker gene is only used for screening purpose. Can you leave the marker gene there? Of course. If you think the marker gene doesn't affect any of your study, then it's okay. You can do this. Then there's another way we can validate this. So it's like we can put the human DNA into our model organism. And here, because we have a GAL4 system, so I use GAL4 UAS. UAS is the upstream activation sequence, and it was bound by the GAL4. Then this binding, if this binding occurs in the cell, then this human cDNA will be activated. Of course, you can direct target the human cDNA, integrate the human cDNA into the fly locus. So you can do a variety of these combinations. Depends on your purpose and depends on the subsequent experimental design. Whatever manipulation you did here using model organism to study human gene, and you can do follow-up phenotypic analysis, molecular studies, and try to understand the mechanism. And hopefully all the subsequent study here will help us go back to human and try to understand the causal mutation of this disease, and hopefully that can help us with the drug development. Okay, so I want to go back two more slides. For this method, I think some of you might think, oh, this is the way Drosophila people would do, but we have a variation or sort of like a different way to apply to other model organisms. So think about you have a gene with phenotypic effect. Yes.
Hello?
Can you hear me?
We lost the voice.
You lost the voice. Okay, can you hear me now?
Hello?
Can you hear me now? Oh, I tried to fix it. Can you hear my voice?
Oh, she can hear me?
I can hear you. Yes.
No.
Okay, we are checking it.
No, absolutely not. I can't. There's the sound. There's no sound coming from Taiwan.
Okay, too bad.
Could be. Is that the mic? I thought originally the sound was coming from this speaker and then it switched to that speaker. No. Maybe changing the microphone.
Okay.
Oh yeah.
Can you hear me now?
Oh no. Sorry. I'm sorry. I wasn't.
Okay, it's not the microphone. Because here we also can hear the microphone. Oh.
Sorry. No.
Let me try.
Yeah.
So can you hear from here? If I change to this microphone, can you hear me? No. So I don't know what we can do, but let me see whether you can hear this. I have a short video for 4 minutes. Can you hear that? Okay, so I hope you can hear me now from Japan side. Can you hear me?
Sorry, Professor Ting, we still can hear you. Is that our speaker?
Yeah, can you check
We
your
lost
speaker?
sound. We can see it, right?
But just to
There's
save
some
time,
connection.
I want to go to the discussion section. So we'll try to fix the issue now and go to the discussion section. So I have two questions. So I think both are not too difficult. The first question is, think about a gene you think it's important and you want to target, and in what organism, and you can think about this. So this is like the first one. And the second question is, there's a lot of discussions on applying CRISPR in human cells. Then I want you to think about what's the limit for clinical application. And the limits could be technical, how difficult it could be, and it could also be ethical. So then, we will have 20 minutes for discussion. Then during this time, we'll try to fix the issue. Okay, so we can discuss now.
Professor Ting, how many more slides do we have? Or no? We can't see the slides either. Maybe it was our speaker, right? Because I didn't hear myself either. Okay, so now we can see the slides. All right. So you suggest we start the discussion?
Yes. Yes.
Oh, there. We can hear you now.
Oh, great. Okay.
I think there's
Good. Actually, don't have to worry because we were just watching a video and I have that link on my slide. So you can watch that 4 minutes video later. Then I have two questions for students to discuss here. One is more sort of like a practical, think about what gene you would like to target and why you want to target, what's the importance. And the second thing is to think about what's the limitation of like in applying CRISPR in human. And the limitation could have at least two parts. One is the technical limitation, how we do how could we do that and how that can really, into the human body. The second could be ethical, what type of gene we shouldn't do that, or what type of cell we shouldn't do, or it's okay as long as we want. So these are something we can discuss. There's many discussions, I think, on the website and some paper already discuss it. But I want you to really, as a group, discuss these two issues. Then we have 15 minutes. Then we'll share what we think. Okay?
all right. Thank you very much. All right. You got it? Oh, you want to watch it again? Can we watch the YouTube video again? We? They did. Yeah, yeah. Would you show that the movie again? Yeah. Thank you. Thank you, Chati. Okay. So you have lots of ideas now. So let's break up for discussion. Eh, Let's do 15 minutes. Professor Ting, shall we answer both questions or pick one of them?
We can just pick one of them. Yep.
One of, all right, thank you. Professor Ting, shall we get started?
So I think this week, should we start from the Taiwan side? Okay, so we'll start from here, then we'll switch to Japan. Yeah, please.
Hi, hello.
Okay.
Hi. Oh my god.
Hi, give us a second. Some people are still talking. Guys, Taiwan team is about to present. Okay. 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 Good. Please
Hi.
start.
Okay, we want to answer the question number two. We think there are many problems if we use this technology on human. Um, to if we talk about technology problem, we don't know how steady the technique is. Maybe Cas9 will off target to the wrong gene and cause more problem on the human. And if we want to cure disease in human body, we can't change the cells, all the cells by this technology. It's too difficult. For example, if I got cancer, I added a cancer cell, but how about other cancer cells? And we have to change all the cells, so it's very, very difficult. And about the ethic problem, we are not God. We can make a human just like God create people. So if we do that, a lot of problem we can imagine will happen. Yeah.
Okay, thank you. So how about we switch to Kyoto side?
Okay. That was a very interesting answer. Thank you. Where shall we start?
All right, hello. So we want to tackle the first question, and we were thinking of um, of cancer in Drosophila. So there's this gene, BAC beetle, in Drosophila, and RAS protein in Drosophila. So RAS is responsible for cancer, and but when we only have RAS, um, and back beetle is inactive, the gene is inactive, you have overgrowth of the cells. So the cells just enlarge, the cancer cells. But when back beetle is active, instead of enlargement, the cancer cells are capable of invading surrounding cells. So we were thinking that we would use the Cas9 system to target the back beetle and see what kind of changes occurs to the cancer cells. Thank you.
Okay, thank you. Next group.
Hi. We talk about question one. We would like to edit OSBP, which is a human gene related to cholesterol transport from ER to Golgi apparatus. OSBP mutant break down intracellular cholesterol transport and cause dyslipidemia, so OSBP is an important gene. That's all, thank you.
Okay, which specific cell you want to edit? You want to edit at which stage? How you apply that to the human cell?
Sorry.
Yeah, that's okay because
Right.
I think think about gene and the importance of gene is okay. But another thing, if you want to do in human, then you have to think about how you want to deliver the system to the cell at a cellular level and which specific stage. For example, during our discussion, we're thinking about the blood stem cell, which we know quite well, and we know how blood cell developed, and and it can be isolated outside the human body, cultured, and we can modify the cell, then put it back to the human body. Something like that has to be really in your plan. Right? Thank you. So next group. Who wants to present?
Okay, our team talk about question number 1, and I don't know it's real, whether it's realistic. It's just hypothesis, and we want to introduce photosynthesis gene family to animal cell, so we don't have to eat. We just get energy from sun bath. So that's it.
I think it's a very interesting idea if I hear it correctly. This group wants to just very creative. They want to put a photosynthesis system into the human cell, so we don't have to eat, then they will be free of hunger. But I think, yeah, even this can be applied. It's a very interesting part. But I think I still enjoy eating, because eating is not only for, in a lot of human population, eating is not only for just not starve, but you enjoy the taste, and there's a culture involved within your meal. So anyway, okay, next group.
I'll answer for question number 2. One problem is off-targeting effect. Since guide RNA recognize complementary sequence, so sometimes nuclease cuts unexpected regions which have similar sequence with objective regions. So mutations are induced via non-homologous end joining incidentally. So systemic influence may occur due to off-target effect. So CRISPR, if CRISPR doesn't work specifically.
Thank you. Next group.
We would like to edit scramblase. So it is one of the enzyme. So scramblase transports lipid molecules inside and outside cell membranes. So one of the lipid molecules, phosphatidylserine and phosphatidylcholine. Phosphatidylserine works as eat-me signal. So we knock down the cell group of scramblase. It will not expose PS or incorporate PCs of phosphatidylcholine. So the gene relates eat-me signal and apoptotic because I think it is important. Thank you. That's all.
But the similar question is which type of cell you want to engineer and how you want to apply to human. Is there an answer for that?
Sorry, I don't understand what you say.
I know the protein you want to see whether I got it right. you want to modify the protein which transport lipids, right? So just assuming you can do that, and what type of cells you would like to engineer, and how would you apply that to the organism level?
So I use serum cells, so B cells or T cells, and the organism is a mouse, maybe mice.
So you want to do this in mouse, is that right? Okay, thank you. Next group.
Hello. So our group also discussed question 1, and we were interested in a disorder known as progeria, which is where there's premature aging, and this results from mutations in a gene called LMNA. So we were thinking of using CRISPR and Cas9 to introduce mutations in LMNA in model organisms such as mice and Drosophila to use it as a tool to study potential treatments and ways to manage the disease.
Yeah. Okay. Thank you. Yeah. This is quite a typical study. Nowadays, you see a lot of paper. They use not only one but 2 model organisms to study the same gene function in 2 systems. Then try to address the question. And I think you will see that more often because CRISPR. One thing I didn't talk about today is it's not limited to model organism anymore. There's many organisms you can apply the CRISPR technology. Okay, thank you. Next group.
Our group also decided to talk about question 1, and our motivation is to develop a new treatment for cancer, and the target gene is not detected, but we hypothesize some kind of cancer marker gene or some kind of marker array that could change the chromatin structure when the phenotype is cancer-like or not cancer-like. And we use the cells are mouse cancer cells or mouse normal cells, and we edit these kind of marker genes and induce apoptosis inducible genes and detect that we could only kill the cancer genes when it was injected. So that's our research, and when the problem is when we need to apply it to human treatment, it is hard to deliver these CRISPR-Cas systems to all cancer cells, including some kind of cells that are transferring into the blood.
So this is really an ideal situation. You can use, so if assuming you can identify specific cancer marker, then you can target. But I think this group, they're also very thoughtful. They think, but how do you deliver it? Because deliver the Cas9, the whole system into tumor will be a very difficult task. But this is something in principle might work. But there's a lot of people also working in this direction, try to think about how we might be able to utilize this machinery. But I think the basic idea is here to target cancer-specific cell and trigger the apoptosis pathway. So all the cancer cell will be sort of like eliminate from the human body. Thank you. Next group.
Hello. We are going to talk about number 2. It's dangerous to apply it to humans, such as to cure the disease. There are several factors. There are several barriers which prevent this application. First, there is off-target. So Cas9 protein causes the region which we do not intend to. And number two, CRISPR-Cas9 comes from virus. So even if it's possible to apply to human, there is a chance that immune system would react to Cas9 and reject it. And in the result, it cannot work or like cause allergy. So for these two reasons, it's still difficult to apply to human. Thank you.
Okay, great. Yeah, I think as the first group here in Taipei also mentioned, one thing I didn't talk about is the off-target effect. Do we know, like, can we estimate? Of course, in the model organism, we are sort of like less worry about the off-target effect because you can always do subsequent backcross or you can do transformation to prove the gene function. But in a human cell, if you target the human cell with off-target effect, and that's something we don't want. But once you did that, it's fixed in the cell and propagate if you deliver into the human body and you propagate. So that's one of the important issue other group also mentioned. The other thing is how effective, so you have to think about what type of cell you want to engineer and whether this engineering will be effective to the whole human body. And this is also some issues. The other one is about the ethical part, whether we want to modify the germline or even in the early embryo, and can we do that? I think also the first group here is also mentioned about the issue. Okay, do we have more group at the Kyoto side? No? Okay. Okay, then thank you for all the very good discussion and presentation. Then I think we will end today's lecture here. Then next week we'll move on something else. Thank you.